This data visualization plots over 75,000 pathogenic human genetic variants currently logged in the ClinVar database. We have mapped the exact molecular coordinates for every single one of these structural errors. But knowing the coordinates does not equal the ability to fix them. Standard Cas9 nucleases relied on double-strand breaks, which trigger a cellular repair process that often results in unpredictable mutations at the target site. Base editors bypassed the double-strand break problem, but they have a limited scope. They are restricted to specific transition mutations, swapping C for T or A for G, and they frequently introduce bystander edits to adjacent healthy bases. This leaves a massive cluster of uncorrectable variants on our map. This remaining territory consists of transversions, insertions, and deletions. These are complex, multi-base errors that earlier precision tools simply cannot address. Take the HBB gene mutation responsible for sickle cell disease. This is a transversion error, a single-letter swap that radically distorts red blood cells. Correcting this requires a level of precision that nucleases and base editors cannot reliably provide. We have the genomic map of human disease, but the field requires a search and replace mechanism capable of rewriting that map without relying on the cell's inefficient homology-directed repair pathways. In 2019, a system emerged that bypassed both double-strand breaks and the requirement for exogenous donor DNA. It is known as prime editing. Based on its biochemical architecture, prime editing is theoretically capable of correcting up to 89% of all known pathogenic variants. It achieves this by physically fusing a modified CRISPR complex with reverse transcriptase machinery derived from the Myeloid-murine leukemia virus. This architecture enables the direct writing of new genetic information into the target locus, treating the genome as a rewritable text. At the core of the prime editor is a Cas9 protein modified so one of its domains is deactivated. This makes it a nickase, capable of cutting only a single DNA strand. Engineers attached a reverse transcriptase domain to this nickase, forming the complete prime editor complex. Standard single-guide RNA is insufficient here. Instead, it requires a prime-editing guide RNA, or pegRNA. The spacer sequence directs the Cas9 nickase to the target locus. The primer binding site physically anchors the DNA. Finally, the reverse transcription template encodes the newly corrected genetic information. The pegRNA operates simultaneously as the genomic GPS, the physical anchor, and the blueprint for the new genetic code. Cas9 cleanly nicks a single strand of target DNA. The nicked 3' end lifts away, hybridizing with the pegRNA's primer binding site. Anchored here, the reverse transcriptase uses the DNA as a primer to read the RNA template, polymerizing the edited sequence directly onto the target strand. Because this polymerization happens directly onto an existing nicked strand, the edit is written without triggering the error-prone non-homologous end-joining pathway. This polymerization creates a branched heteroduplex structure with an unedited 5' flap and a newly synthesized 3' flap. Cellular endonucleases automatically cleave the unedited flap. The edited 3' flap then ligates into the genome, leaving a base mismatch between the edited top strand and the unedited bottom strand. The cell's mismatch repair system might detect the anomaly and erase the new edit. To prevent this, PE3 uses a secondary guide RNA to nick the unedited strand. This forces the cellular repair machinery to erase the unedited DNA and copy the edited top strand, safely resolving the mismatch. Prime editing succeeds by actively manipulating the cell's own quality control machinery to permanently lock the edit in place. We are now seeing the first clinical application of this biochemistry with the PM359 trial. The clinical target is chronic granulomatous disease, an immune disorder caused by the del-GT mutation in the NCF1 gene. The treatment utilizes an ex vivo delivery mechanism. Autologous stem cells are extracted from the patient and electroporated with the prime editing machinery. The prime editor successfully inserts the missing bases to correct the mutation, restoring myeloid function before the cells are returned to the patient. This milestone proves prime editing can achieve disease-modifying cellular correction in humans without the genomic instability associated with traditional double-strand breaks. The primary physical constraint holding the platform back from broader applications is viral packaging capacity. The industry-standard AAV vector has a cargo limit of 4.7 kilobases. The prime editor construct is massive, measuring roughly 6.3 kilobases. The full fusion protein is simply too large to fit inside the standard delivery vehicle. To solve this, researchers developed the dual AAV split PE system. By utilizing split inteins, the complex is delivered in halves, allowing the protein to reassemble itself inside the target cell. Alternative delivery vectors, such as engineered virus-like particles and lipid nanoparticles, are also in development to bypass viral limits entirely. The development of these delivery systems will determine whether the platform can move beyond blood disorders. If the editor can reach internal organs, its versatility makes it a candidate for correcting a significant portion of the